In today's practical, we will be analyzing the amount of phosphate in cola samples. Compounds absorb light of different wavelengths due to their structure. The absorbance of the sample is governed by the concentration of analyte in the sample and by the path length over which the sample absorption occurs. A spectrophotometer is used to measure the absorbance. In this experiment, molybdeno-vandophosphoric acid is synthesized from molybdenate, orthophosphate, and vanadinate, in which a yellow color is formed. The color formed by the complex is dependent on the concentrations of the reagents and the acidity of the solution. The aim of the experiment is to determine the amount of phosphate in cola samples by complexing the phosphate to the synthesized complex and by measuring the absorbance. You are provided with 1000 ppm of phosphate solution. You will need to transfer 5 ml of this solution using a pipette into a 250 ml volumetric flask. This would provide you with a 20 ppm of phosphate solution. Once the phosphate solution is added to the volumetric flask, you will then make it to the mark using the deionized water. For this, you can use a beaker instead of your wash bottle. Ensure that you do not overshoot the mark. And once you are close enough to the mark, you will use a dropper to get the meniscus on the line. Once you've done that, you will stop at the flask and invert it 10 times. This is your 20 ppm phosphate standard solution. You are provided with a cola sample. You will need to take 10 ml of the sample using your pipette and transfer it to a 100 ml volumetric flask. You will thereafter make it to the mark using deionized water. Do not overshoot the mark. Once you get close enough to the mark, you will use a dropper to make it to the mark. Ensure that the meniscus sits on the mark. Thereafter, you will invert your solution 10 times and into 3 clean 100 ml volumetric flasks, you will add in 5 ml of your Coke solution. Once the solution is added to your 3 volumetric flasks, do not make it up to the mark. You are now ready for the next part of the experiment. You will prepare burette using your 20 ppm solution of phosphate. Thereafter, you are provided with 6 100 ml volumetric flasks, which you will label 1 to 6. In the first flask, you will not add any of the solution you've prepared. Into the second flask, you will then add in 8 ml of your phosphate solution using your burette. Into the third flask, you will add 16 ml of the solution. The fourth flask, 32 ml of the solution. To the fifth flask, you will add 40 ml of the solution. Once these solutions have been added to your flasks, you will set them aside and prepare for the next part. You are provided with 3 solutions: a nitric acid solution, a vanadinate solution, and a molybdenate solution. You will prepare your burette with the nitric acid solution and then add your nitric acid to each of the 9 flasks that you've prepared. 10 ml into each flask. You will then clean out the burette and add in your vanadinate solution. Thereafter, 10 ml into each of your volumetric flasks before cleaning out the burette. Lastly, you will add your molybdenate solution into the burette and add 10 ml to each of the flasks before cleaning out your burette. Thereafter, you will make all your flasks up using deionized water. You will have 6 standards and 3 samples of cola. You should note an intensity increase between standard 1 and 6 in terms of the yellow color. Your samples should essentially be the same color in intensity. You should have 6 solutions increasing in intensity of the yellow color. These are your standards. You should also have 3 cola samples which have the same intensity of the yellow color. Once your solutions have stood for 30 minutes, you will obtain a cuvette. The cuvette has 2 sides, a frosted side and a clear side. You will only hold the cuvette on the frosted side. You will take a piece of tissue and wipe down the clear side to remove any fingerprints or dust. Thereafter, you will take a clean beaker and your distilled or deionized water and rinse out the cuvette 2 times. The third time, you will fill the cuvette up to nearly full. Thereafter, you will transfer it into your spectrophotometer and have the frosted side facing you. Once it's in, you will close the lid and zero the instrument. You need to look at the screen to ensure that the instrument is set at 400 nanometers and then press 0 to blank the instrument. Once your solutions have stood for 30 minutes, you will obtain a cuvette. The cuvette has a frosted side and a clear side. You will only hold the cuvette on the frosted side. You will get a piece of tissue and wipe down the clear side so that you remove any fingerprints or dust. Thereafter, you will rinse out the cuvette using some distilled or deionized water. You will then fill up the cuvette with your distilled water to almost full. Thereafter, you will transfer it to your spectrophotometer, frosted side facing you. You will place the cuvette into the box with the frosted side facing you. You will then close the cover. You will ensure that the instrument is set at 400 nanometers before zeroing the machine. This will blank the machine with your distilled or deionized water. You will then empty your cuvette out, mix your first solution, add some of the solution to your cuvette, and rinse your cuvette out 3 times. Thereafter, you will fill your cuvette to nearly full, wipe down the sides, and you are ready to place this into the spectrophotometer. You will place the cuvette with the first solution into the box and record the reading to 3 decimal places. Thereafter, you will remove the cuvette, rinse it, and repeat this with the rest of the solutions. You should see an increase in the absorbance values from solution 1 to 6. Thereafter, you should have a fairly constant value between the 3 cola samples. From here, you have got your data to plot your curve and determine the amount of phosphate in cola. Data analysis. You have to calculate the mass of phosphate in micrograms in each standard. Plot the mass of phosphate versus the absorbance. Remember to use your reagent blank. Calculate the mass of phosphate in each replicate sample. Calculate the percentage of phosphate in cola as percentage mass by mass and ppm of phosphate. Remember to include your stats analysis in your critical discussion of results.